Hello, my name is Pauline Mouches. I'm a PhD candidate at the University of Calgary in the Biomedical Engineering department, and I'm also a CONP scholar. Today, I'm going to present my research project, which is about studying healthy brain aging. So the motivation for my project is that structural changes in the brain occur due to natural aging with, for example, atrophy of the brain tissues. But some of these changes also occur in the case of neurological diseases. And so the idea is that a better understanding of healthy brain aging is needed in order to better diagnose neurological diseases at an early time point. So the goal for my project is to use advanced image processing and machine learning techniques to combine imaging and non-imaging data and give new insights of brain aging patterns. The database that I've been using for my project is made of about 2,000 adults aged between 20 and 80 years old. And for each of them, I have access to two kinds of imaging data, which include magnetic resonance imaging on the right here, on the left here, sorry, where we can clearly see the brain tissues, and also magnetic resonance angiography, where we can see the brain arteries in white here. So I also have access to cardiovascular risk factors information for the patient in this database, and this factor include, for example, body mass index, blood pressure, or smoking status. In terms of computational methods, the idea is to identify relationships between the imaging and the non-imaging data, which in this case are the cardiovascular risk factors. So there's mainly two main approaches that I've been using, with the first one being using extracted metrics from the images and finding relationships between these extracted metrics and the cardiovascular risk factors using machine learning models. And the second approach consists in using the images directly in combination with deep learning models in order to, again, identify these associations. These are the key results for my project. So the first part consisted in studying the influence of cardiovascular risk factors on brain arteries. And in this work, we used extracted metrics from the angiographic data sets. So the idea here of extracted metrics is to use, for example, brain arteries, diameter or density. And then we use multivariate regression analysis to find the associations between these brain artery related metrics and the cardiovascular risk factors. Secondly, I worked on the biological brain age prediction topic, which has been of high interest recently. And the idea of this topic of biological brain age prediction is to use brain images to predict or estimate the age of subjects using deep learning models. So in my case, I've been using both imaging modalities, and I've been furthermore implementing explainability methods to understand which parts of the image, so which regions of the brain, were used by the model to make the age prediction. And that's actually what we can see on the image here, where the arrows highlight the parts of the brain that were used by the model to predict the age. So in conclusion, we can infer that these brain regions are undergoing high changes with aging. And to finish, still on the biological brain age prediction topic, I've been implementing an invertible model. So the idea here again is to use the image to predict the biological brain age. But in the case of an invertible model, we can go also in the other direction, which is going from a specific age to reconstruct a simulated image corresponding to that age. And that's what is shown here as example in the image, where we have 3 different ages, and below the ages you can see the simulated images corresponding to each age. And so the main advantage of this kind of new techniques is that we can directly visualize the age-related changes on the images. During my project, I've been trying to contribute to the open science community. And to do so, I've been mostly following these 3 bullet points here. The first one consisting in targeting open access journals and conferences publishing proceedings online. The second point is, to my opinion, very important, is to make the implemented codes available and understandable so that any researcher can reproduce our methods. And finally, the last point is to write reproducible research by providing implementation details in the publications, again, so that any researchers can easily either reproduce the method or adapt it to a specific application. So with all that said, I would like to thank CONP for funding my research and for providing me with great resources about open science. Thank you.